All right, let's get right into it. We tend to think of our cells, especially the ones that are genetically identical, as this perfectly uniform army, right? All of them just marching in lockstep. But what if they're actually more like a bustling crowd, where every single individual has its own little personality? That's exactly what we're going to explore today, the wild, surprising individuality that's hidden inside every single one of us. And to see why this matters so much, let's start with a really high-stakes question. Think about cancer treatment. A therapy can wipe out, say, 99% of a tumor, but a few stubborn cells just survive, and then they cause a relapse. Why is that? Why do some of those cancer cells make it when others, with the exact same DNA, get destroyed? Well, the answer, and this is the really fascinating part, isn't always hiding in their genes. The answer lies in this incredible hidden feature of life, cellular individuality. Now, scientists have a formal name for this, of course, non-genetic heterogeneity. What this really means is that cells with identical DNA can react in completely different ways to the exact same signal. It's kind of like a group of identical twins hearing the same piece of news. One of them might laugh, one might cry, and another just shrugs it off completely. So let's pull back the curtain on this hidden rule of biology. How does this individuality actually work, and where does it have the biggest impact on our lives? And believe me, this isn't just some weird biological curiosity. It has huge real-world consequences. It's the very reason for drug resistance and cancer. It's what governs how our tissues develop, deciding if a cell is going to become part of your heart or your skin. It even affects how tiny microbes handle stress and how viruses manage to take hold in our bodies. This variability is literally everywhere. Okay, so to really get where this individuality comes from, we've got to take a little journey inside the cell and check out its super complex internal communication network. So think of a cell's signaling pathway as its internal chain of command. A message, maybe it's a hormone or a drug molecule, shows up at the cell's front door. The pathway then carries that message all the way inside to the nucleus, which is basically the cell's control center, to trigger a very specific action, like divide now or time to die. Let's look at a real example, this TGF-beta pathway. It all starts when a signal arrives at the cell's surface. And that one touch kicks off this whole chain reaction inside, like a line of dominoes falling. Key proteins get activated, they team up with other proteins, and then the whole package travels to the nucleus. And what's their final job? To act like a light switch, flipping specific genes on or off. It's the super precise cascade of events that tells the cell exactly what to do next. So the big question is, how in the world can scientists actually see these subtle little differences between individual cells? I mean, they're tiny. This is where modern technology basically gives us a kind of crystal ball to see what's going on inside. The old way of studying cells, it was a lot like listening to the roar of a crowd at a stadium. Scientists would just grind up thousands of cells together and measure the average response. But the new way, using single-cell analysis, that's like having a microphone on every single person in that crowd. You don't just hear the roar anymore. You hear every individual voice, every unique reaction. It's a total game changer. And here's why that is so important. That old averaging method is seriously flawed. Imagine a population where every single cell makes a clear digital on or off decision. Well, if you average all those sharp responses together, what you get is a blurry, gradual-looking result. You completely miss the fact that the decision being made at the single-cell level was actually black and white. But today, scientists can do way more than just listen in. They can actually create a virtual cellular crowd right on their computers, which lets them run experiments that would be completely impossible in the real world. Let's see how this incredible tech works. They pull this off using something called a mathematical model. Often, it's a set of what are called ordinary differential equations, or ODEs. Now that sounds complicated, I know, but the absolute best way to think about it is like a flight simulator, but for biology. It describes all the chemical reactions happening inside a cell, letting scientists test any what-if scenario they can dream up. So how do you actually build one of these virtual populations? Well, first you create a super detailed model for just one average cell. Then you make thousands of virtual copies of it. And here's the magic step. For each and every copy, you slightly change the starting amounts of the key proteins, just like the natural variation you'd find in a real population of cells. And then you just hit run and watch how this diverse group responds to a signal. And the results are stunningly accurate. As the researchers who actually developed this method put it, they got an in silico cell population with realistic properties. So this isn't just some random guess. It's a simulation that actually mirrors the complex, messy behavior of real biology. So what's the big revelation from these models? Well, it turns out that you can group these supposedly identical cells into distinct personalities. Some cells are just non-responders. They completely ignore the signal. Others are transient responders. They react, but only for a short time. And then you have the sustained responders, the ones that keep that signal going. Three totally different fates, all from one seemingly identical starting group.
Okay, so cells are individuals, and we can model them. But why does all this cellular chaos really matter? What is the actual real-world payoff of knowing this?
This is where it all comes together. By understanding and being able to predict this variability, we can finally move beyond one-size-fits-all medicine. We can design drug combinations that don't just target the average cancer cell, but also hunt down those few resistant individuals that cause a relapse. We can get so much better at predicting how tissues form, and maybe even learn to guide cells for things like regenerative therapies.
And all of this leads us to one final really powerful question. If we can truly understand and predict the behavior of a single cell, could we one day program its fate? Could we literally tell a cancer cell to self-destruct or instruct a stem cell on exactly what kind of tissue to become? The ability to finally see the individual in the crowd is opening up a future that we are only just beginning to imagine.